So I'm going to introduce the third speaker, and he's the last speaker telling us about translational control. I'd like to welcome Zhenguo Zhang. He's going to tell us about translational compensation for aneuploidy.
Okay, I would like to add my gratitude to the organizer and the session chairs for giving me the opportunity to present my work here. So specifically, my work are very computational, but let's hide the technical details for the time being. So I focus on the biological questions. So, so the aneuploidy is a process. So that's the chromosome 1 is people know well know. So this is in the case one chromosome is increased. In this case, you get 3 copies of one chromosome. This is called chromosome aneuploidy. And there are cases called segmental aneuploidy at the bottom. So those are regions, genomic regions get duplicated or get deleted. So that's the second case is what I focus on today. So the aneuploidy have a lot of biological consequences, and in some cases they can help the cell to adapt to the new environment. And more often, people know they relate to the cancers, and also in general, they reduce the cell fitness. So all of these are related to the effect of the aneuploidy on gene expression. As a case for the S2 cell line, this is the most popular cell line using Drosophila. The y-axis is copy numbers for each genomic region, and also represented by different color, for example, at the top. And so most genes regions have the 2 copies, but their regions have more than 2 or less than 2. And so the question is how these genetic changes will impact the gene expression and the cellular level as a whole. So it's already known that the gene expression at the MI level can be compensated. For example, so I think I need to, because this is, let's see, I don't know how to get this. So this is a box plot. So this middle line is a median value. This we should focus on in this talk. So that's you will see a lot of this box plot in my slides, and those are the top. For the box for the top boundary and the bottom boundary are given the 75 percentile and 25 percentile. So these are statistical details. So let's focus on median value at this moment. As these 2 dashed lines give the expected gene expression for the gene based from expected gene expression based on gene dosage. If the genes has no compensation happening at all, so you expect the median value will match those 2 dashed lines. But in this case, for the duplicated genes, you can see this median value is lower than the dashed line. This means there's compensation. So, but the compensation could happen in many ways. So the transcription MIDK can affect the steady state MI level. And further down, we can also study how translation protein degradation affects the gene expression and the protein level. And the translation is what I will focus on today. So my outline is I'll give you a brief introduction how we use the genome-wide technology to measure translation, and then study translation rates in S2 cell line. I'll also give a genome-wide analysis of sequence features which may contribute to this regulation of translation, and also finally I'll show some new data in the KC167 cell line. This is another cell line used very often in Drosophila. So I think everybody knows the translation rate term. I just want to make it clear we are on the same page. This is defined as the number of proteins synthesized per MIA copy per time unit, which is somewhat measured by the number of ribosomes on a certain MIA. This is the basis to use ribosome footprint technology to measure translation rates. So this technique is very like chip-seq, but the difference is that we can get the MI fragments protected by ribosome, when we sequence this population, we get the abundance of the ribosomes for MI population. And similarly, we use a regular RNA-seq, we get abundance of MIs. The ratio of these two, we call it ribosome density, and this is give you an approximation of the measurement of how many ribosomes are translating on given MRAs. So the public data we use with S2 cell line. In addition, we use another 2 samples which are embryos and also another cell line as a control. So this is the box plot again. So here there are 3 groups. So the middle one is a normal group which have normal number of copy genes. And the left one, which we represent with pink, and which are the genes have actually lower copies, means depletion, and the right one with blue will get our genes with increased copies. You can see compared to the middle one, so when the genes decrease their gene copies, they will have higher translation rate, and you will see the opposite for the copy number increase genes. So this is for the X chromosome, and all chromosomes show the same one. Okay, there are 2 possibilities for this explanation. The first one I already give explanation, which is the explanation I already give you. This means this kind of translation compensation. But it's also possible that this might be a property of this copy number changed genes. So to test that, we do the same analysis in the embryo data. So because these are the genes are in each group are exact same as the previous analysis, we don't see the difference. This means they are not the features, this kind of compensation is not a feature of genes themselves. Consistent with this, we analyze the sequence features which could affect the gene translation, and nearly all the features are equivalent for the gene copy number change genes and the normal genes. And except for one case, which is the 5' UTR but the red one now plus minus this is expected result for the copy number decreased genes and copy number duplicated genes. What we observed, and this are inconsistent with the prediction based on 5' UTR. So we think together these are not due to sequence feature of those genes, and it might be possible that these observed differences could still be due to different functional categories of the genes. So in other words, here I use a different shape in a protein complex to represent different genes, and the different color to represent their copy number status. So the pink one are the copy number decreased genes, and the blue one are the copy number increased genes. So we can calculate ratio. So for the gene within a protein complex, this ratio, if there's compensation happening for the depletion case, the ratio will be greater than 1. So this using protein complex as a way to control the functional consistency, because the genes in the same protein complex are expected to involve in the same process. And similarly, so if for the copy number increased gene, if there's a compensation happening, they will expect a lower, smaller ratio than 1. So this is exactly the case we observe for S2 cell line. So it's interesting to know, we know that compensation at the AMI level, also now the translation level. It's interesting to know at the final protein level, when you look at the product at a gene, whether they will reach say equivalent level. It doesn't matter which gene copy, how many copy gene numbers for each gene. So so the first panel is for MIA compensation. There's a weak compensation, and the second one is translation compensation. And when we put all these together, and finally at the translation level for each gene, we don't see any difference anymore. So this means fully compensation too. So now, we want to know how this phenomena is, how general it is in different cell lines or tissues. So this is limited by what data we have now. So, but luckily we can get the data for the KC167 cell line. So this is different from the S2 cell. So this cell line is represent a female genotype, as also tetraploid. So a similar figure as the previous one for the S2 cell. So surprisingly, we don't see any compensation happening for this translation compensation for this cell line. So this is very surprising, also very puzzling. To understand this, so we try to compare the sequence, the features of these two cell lines. So you can see the S2 cell line is a male genotype, KC cell line is a female genotype, but we don't know how this contributes to the different response of translation to aneuploidy. And also, both cell lines come from embryo. And finally, when we analyze the sequence features by comparing the copy number altered genes between two cell lines, we don't see much difference in sequence features, also functional categories. But so this is really puzzling. But when we look at other literature, so there are already a lot of studies to study the compensation at the MI level. So I give you an example. So even at the MI level, the dose compensation is not a general phenomenon. So S2 cell line is in the first panel, as already shown, it is partially compensated. But for another cell line, this cell line has no compensation at all at the MI level. And to summarize that study, so for analyzing 8 cell lines, we can see there are 2 cell lines have no compensation at the MI level at all, just like the second panel. And there are 5 like the first panel. And there's a complete compensation at the MI level. So I want to conclude that the dose compensation of aneuploidy can happen in certain cell lines or certain conditions, which can happen at both the MI translation level. And this might be contributed by 5' UTR difference. And because there's no general compensation happening for all the cell lines, so I speculate that the compensation, whether it happens and the strength of the compensation depend on the cellular context of each cell line or each gene. So I think we should go to a systems level analysis to really answer why they are different. So finally, I want to thank my lab members and for my advisor support in my research and my funding resources and Genetic Society for my travel award. Thank you. I'm happy to take questions.
So I have 2 questions, both of which may be slightly naive. How stable is the ploidy of the S2 cell line? So from lab to lab, does it differ, or is it that it's achieved this ploidy and is now stable?
I think it's a very good question. So I don't think anybody already studied that. But one piece of information I could provide you is the aneuploidy is very heterogeneous for the cell population. So what I presented to you is 4 copy. For each cell, there's 4 copies of autosome, 2 copies of X chromosome. But which region goes 4, which region goes 2, it's very heterogeneous for the cell line. So different cells have different aneuploidy status. Yeah, it's very heterogeneous.
And just a quick follow-up related question. Presumably that aneuploidy has been selected for during the establishment of the cell line. So if it's being selected for, what is it doing if the cell is then effectively fighting to undo it?
I think it's possible. So this kind of speculation is possible, and based on, but I'm not sure this really provides advantage for the cell to maintain in the lab or somewhere else. So, but I think it's another way could be at the main we get the cell line from beginning. The founder effect may be already there. So some cell line at the beginning already have aneuploidy situation there, but based on the study and literature, they said at least maybe 60% of the aneuploid status is acquired during the cell line evolution. And I don't know this advantage, just like the cancer cells, this might provide some advantage, but I think more studies are needed to resolve that question.
Okay, could you find any functional gene groups that actually have better compensation at translation level?
Could you repeat that question again?
Are there genes that have better compensation in terms of with specific functions or
I didn't go to the details for each gene. So my purpose is to see at the cellular level how the cell responds to this kind of large-scale changes. So I will be happy to talk details after this meeting because the time is approaching. Thank you.
Okay, thank you again, Zhenguo.
Thank you.